We're honored to have Dr. Ann Zajicek provide today's lecture. Dr. Zajicek is a board-certified pediatrician and a pediatric clinical pharmacologist who currently serves as Deputy Director of the Office of Clinical Research at the NIH. Ann received a bachelor's degree in pharmacy from Duquesne University and a doctorate of pharmacy degree from State University of New York at Buffalo. She then completed postdoctoral fellowship training at St. Jude Children's Research Hospital. After that, she served as assistant professor at the University of Colorado School of Pharmacy and a clinical pharmacist at National Jewish Hospital. In 1991, Ann entered medical school at the University of Pittsburgh. In 1998, completed residency in pediatrics at Children's Hospital in Pittsburgh. She practiced primary care pediatrics for 2 years and then continued her training in pediatric clinical pharmacology at Stanford University. She subsequently joined the FDA's Office of Clinical Pharmacology and Biopharmaceutics. In 2003, she joined the National Institute of Child Health and Human Development. We know you will enjoy Dr. Zajicek's lecture.
Good afternoon. I'm Dr. Ann Zajicek. I'm a pediatrician and clinical pharmacologist, and I want to talk to you today about pediatric drug development. For disclosure, I have no conflicts of interest to disclose, and the presentation reflects my views only, not those of the NIH or the U.S. federal government. Topics I would like to include are the definition of pediatric drug development, some background legislation, extrapolation, outcome measures, elements of a drug development plan, and the need for infrastructure. The definition of pediatric drug development sort of comes in two flavors: the development of an already marketed drug for a pediatric indication, which is either the same as the adult indication, using extrapolation of efficacy with new pediatric dosing and safety information, or a different pediatric indication, again also requiring new pediatric dosing and safety information, or development of a new drug for a pediatric indication. One of the complexities with developing drugs for children include the complexity of the fact that you're dealing with minimum four populations. The neonates, 0 to 28 days, and that doesn't even include the preterm neonates who are younger, up to as early as 23 weeks gestation. An infant, 1 month to 1 year of age, child 2 to 10 years, adolescent 11 to 17 years. Now, we would not be talking about pediatric drug development if it weren't for some legislative information. This is a slide from the 1906 congressional record showing the state of patent medicines in the United States. So at about this time, there were an enormous number of easily available compounds with names like Hood's sarsaparilla, Payne's celery compound. One in particular that was quite popular was the Mrs. Winslow's soothing syrup. These were patent medicines, again, that had proprietary knowledge of the compounds in them that were freely available. And you can see that they contained chloroform, alcohol, marijuana, huge amounts of alcohol. And the one in particular that created most problem was Mrs. Winslow's soothing syrup, which was advertised for colic and for teething pain, which contained morphine. Unfortunately, there were a good number of infant deaths from many of these patent medications. So this was up to about 1905, 1906. There were these deaths from patent medications. In 1936, sulfanilamide, which was a sulfa antibiotic, very useful for strep throat, but came in a capsule formulation. And so a pharmacist at Massengill decided to dissolve it in something that would taste good. And he picked antifreeze, diethylene glycol, which ended up in the death of 106 patients, mostly children, and the suicide of the compounder. And in 1961, the thalidomide disaster caused limb deformities. Again, luckily, thalidomide was not approved in the US. So as a result of basically these pediatric disasters came regulatory acts. So in 1906, the Pure Food and Drug Act, which required that labels of food and drugs must truthfully identify the contents or be pure. 1937, the Federal Food, Drug, and Cosmetic Act stated that drugs needed to be safe. And in 1938, actually, the FDA was created. And in 1962, the Kefauver-Harris Amendment stating that drugs must be effective for their labeled indications. Now, briefly following this, there was an interesting commentary by Harry Shirky, who was a pediatrician as well as a pharmacist, stating that by an odd twist of fate, infants and children are becoming therapeutic or pharmaceutical orphans. In other words, drugs are being developed for adults, not for children. And so therefore, if children were using these drugs, they would be used off-label. So in other words, being used for indications that were not in the labeling or the package insert. So is this a bad idea? Is off-label use bad? The downside is that every patient is an N of 1 experiment. There is no data accrual to learn about safety or efficacy. There's no data accrual to learn about dosing and whether the dose should be scaled from the adult dose, by weight, by body surface area, and so on. So the goal from the FDA has been to try to attempt to get pediatric labeling. So how do you do that? So the three things that are required for a drug label are a dose, safety, efficacy. Should the dose be exposure matched from adult data? What are the short and long-term adverse events? And in terms of efficacy, does the drug improve how the patient feels, functions, or survives? However, again, going back to the diverse patient populations, we're dealing with neonates, infants, children, and adolescents, and all the complexity of those different age groups. So FDA has made several attempts to add pediatric labeling. The first one was the 1994 pediatric rule. So the feeling was that the pharmaceutical companies likely had pediatric data in their files, and the request was, would they forward that data to the FDA to improve pediatric labeling? That was not successful. In 1997, the feeling was that perhaps a carrot for the pharmaceutical industry might help, and the FDA Modernization Act was passed in 1997. 1998 Pediatric Rule, 2002 Best Pharmaceuticals for Children Act, otherwise known as BPCA, and the 2002 Pediatric Research Equity Act, or PREA. So the 1997 FDA Modernization Act gave 6 months of additional marketing exclusivity that would be granted by the FDA if pediatric clinical trials, which were performed according to some stipulations of the FDA, were completed, but not required to show a positive effect. So just the fact that the studies were done and were acceptable to the FDA review division was enough to grant the manufacturer an additional 6 months of marketing exclusivity, which could be fairly lucrative depending on the product. In 2002, the Best Pharmaceuticals for Children Act was passed. This continued the exclusivity provision of FDAMA, the additional 6 months marketing exclusivity. But what was noted in the ensuing time between 1997 and 2002 was that many marketed drugs do not have remaining marketing exclusivity. They're old drugs, they're off patent, and an additional provision included a role for the National Institutes of Health, specifically the Eunice Kennedy Shriver National Institute of Child Health and Human Development, to prioritize therapeutic areas and medications in need of improved labeling, to sponsor pediatric clinical trials, and to submit data to FDA for consideration of labeling. This legislation was passed in 2002, reauthorized in 2007, 2012, and last month in 2017. And I've been involved with this program since 2003. In 2003, the Pediatric Research Equity Act, or PREA, was passed. This was a codification of the 1998 rule, which had been struck down in court previously, that was a requirement to perform pediatric studies if it was anticipated that the drug would be used for the same indication as adults. Now, the ringer here was that if the drug was used for, let's say, prostate cancer, breast cancer, in other words, that there would have to be a PREA waiver and the drug studies would not be performed in children. Now, this was amended last month in August of 2017, and PREA also applies if the molecular target of the drug is substantially relevant to pediatric cancer. This is only for cancer, but it was very important for the cancer population. So just in terms of what's what here, so we have older drugs with some remaining patent protection or marketing exclusivity to be covered under PREA. Newer drugs for the same indication or the same molecular target for pediatric cancer would be covered under PREA, and the old drugs which did not have any patent protection would be referred to the NIH BPCA program. Okay, so in planning a pediatric study, the first question you have to ask yourself is, can the indication of efficacy be extrapolated from adults to children? Then you need some sort of method to select a dose and monitor safety. So the extrapolation policy at FDA is very useful. So if the course of the disease and the effect of the drug are sufficiently similar in adults and pediatric patients, then extrapolation can be applied. Does the current adult condition or indication apply to children in terms of disease mechanism and the disease course? If the answer is yes, then only dosing and safety need to be shown for consideration of FDA approval. If the answer is no, that there cannot be extrapolation, then the full 3 studies of efficacy, dosing, and safety need to be performed. Okay. One of the main issues about doing pediatric drug development is the problem with pediatric outcome measures. So outcome measures in children in the short term include how the patient feels, functions, or survives in terms of disease resolution or improvement in clinical status. Long-term effects are also important in children in terms of growth and development, and this becomes complicated if studies require following children for 2 years, 4 years, into grade school, and so on. Second question, are there pediatric normal values for outcome measures? And I will show you a few slides where there are not pediatric normal values. Are the outcome measures accepted by the medical community and the Food and Drug Administration? And I just want to state here briefly that it's important if you're doing pediatric drug development that you should be having conversations with the Food and Drug Administration, the review division responsible for your drug, to determine if you're on the right path. And does the outcome measure that's typically accepted make sense in a pediatric population? Okay, I wanted to point out two terms just to clarify some language we'll be talking about. Biomarkers. A biomarker is a defined characteristic that is measured as an indicator of normal biologic processes, pathogenic processes, or responses to an exposure or intervention, including therapeutic interventions. And some common examples which we toss around constantly but don't, I think, generally think too clearly about them are cholesterol, blood pressure, serum creatinine, creatinine clearance. So we assume that when we talk about serum creatinine, creatinine clearance, we're talking about kidney function. When we're talking about cholesterol, we're looking at a cholesterol level as indicative of your chance of having a heart attack or a stroke. And this is in comparison to the term surrogate marker. And I thought this was really well stated by Rusty Katz, formerly of the FDA. The primary difference between a biomarker and a surrogate marker is that a biomarker is a candidate surrogate marker, whereas a surrogate marker is a test used, taken as a measure of the effect of a specific treatment. Okay. Now, these are a couple of tests that have worked very nicely in adults but have not functioned well in the neonatal population. The first one is the 6-minute walk test. This is accepted by the Food and Drug Administration as an indicator of functional capacity. It's used primarily in adults to measure cardiopulmonary function and in adult pulmonary hypertension as a biomarker, of reduction in pulmonary hypertension and improvement in function. So you have a patient walk as far as he or she possibly can in 6 minutes. That's a 6-minute walk test. However, neonates cannot walk, and so obviously this is not applicable to neonatal pulmonary hypertension, so we're back to doing invasive cardiovascular monitoring. Another term is blood pressure. This is a surrogate marker for survival of adult hypertension. So if a drug lowers blood pressure, typically it can be approved without following patients all the way toward mortality. However, we did an interesting study with the Neonatal Research Network, and the question was surrounded itself with dopamine. So dopamine is used as a pressor agent to increase blood pressure in adults. The Neonatal Research Network was doing a study, a factorial design of hydrocortisone and dopamine. 366 infants were screened, 10 were enrolled, which was not good. The issues included eligibility and consent. But as a secondary issue, the real problem was about what did low blood pressure mean in a neonate. So how was the blood pressure measured in the neonatal intensive care unit? Have these methods been standardized or validated in this population? What is a normal neonatal blood pressure at a given gestational or postnatal age? And the answer was that there were no normals. What is the different definition of hypotension? Is it just the number? Is it tissue perfusion? Is it shock? Is it lack of urine output? What is the clinical endpoint in the treatment of hypotension, and how is this endpoint measured? So all these questions came up from a very simple question of, was dopamine going to increase the blood pressure? And at the end of the day, it appeared that dopamine actually was not effective in increasing the blood pressure in any of these measures. So quite interesting. This is a slide which is very interesting about reliability of height measurements. This is a very simple study looking at different ways of measuring the height of children in an office setting. So they use stadiometers, rulers, and a wall chart. And you can see the problem here is that the stadiometer did a really nice job of measuring what we are hoping is the accurate height of the child. But the wall charts, which are commonly used in pediatric practice, show values all over the place. So if you're trying to do a clinical trial and you're relying on an outcome measure of height, you have to make sure that you know how it's being measured and that you're training the people who are doing the height measurements to actually measure the outcome measure accurately. Okay. This relates to dosing, but I wanted to make mention of the fact that frequently with pediatric studies, there is a lack of oral pediatric formulations that are swallowable, palatable, and that have dosing accuracy for very young children. And these are a few examples that we've encountered during some of our trials. The first one is a split tablet of baclofen. We had a clinical trial of baclofen. The tablet strength in the United States is 10 milligrams. The tablet is not scored. So if you cut it in half, you're not going to get half of the drug substance in half of the tablet, which is a little known fact. So if the tablet's not scored, you can't cut it in half and get half the dose because the active compound is not dispersed within the tablet. In the upper right-hand corner is an Accutane capsule. So Accutane is used as standard of care for children with neuroblastoma. Unfortunately, the peak age of neuroblastoma is 2 years of age, and 2-year-olds cannot swallow these capsules. So they have to be chewed or squeezed onto food. The problem is that the chemical substance in Accutane is 13-cis-retinoic acid, which isomerizes in light to all trans-retinoic acid. So in other words, if you decide to squeeze it onto food and it's sitting in light, it's probably the patient is not actually getting the drug compound you want them to get. Third example is hydroxyurea. The National Heart, Lung, and Blood Institute has been doing a trial for several years looking at the efficacy of hydroxyurea for children with sickle cell anemia. The issue here is that the most recent trial, the Baby Hug trial, was dosing children 9 months of age to 17 months of age. So in addition to the dose being wrong, no 9-month-old could possibly swallow one of those capsules, and so the drug had to be reformulated into a liquid. So there are many formulations problem. It's something to keep in the back of your mind when you're starting on pediatric drug development, that you need to come up with a product that can actually be taken by the child. Now that answers the swallowability problem and the dose accuracy problem. The other issue is palatability, and that's one of those terms that doesn't have a clear meaning, but I suppose if you could get the child to take it more than once, that would probably answer the palatability question. Okay, so I wanted to give you a concrete example of a drug development program, and I wanted to talk about meropenem. Meropenem is a broad-spectrum carbapenem antibiotic. It's labeled for complicated intra-abdominal infections in patients older than 90 days of age. The labeling gap is in premature infants, neonates, and infants less than 90 days of age. We received a written request that had been declined by the new drug application holder. And the main question was a safety question. Is meropenem associated with an increased incidence of seizures in neonates? There was some indication that imipenem, another drug in that drug class, did increase the incidence of seizures. So the question was this a problem in meropenem as well? Okay, this is a picture of about a 500-gram, very sick, intubated neonate in the intensive care unit, just to frame what the issues are with treating premature neonates of this size. So in terms of the study plan, there was agreement with FDA on the use of extrapolation for efficacy, making the assumption that these severe, complicated intra-abdominal infections were similar to neonatal, necrotizing enterocolitis. We were directed to perform dosing and safety study in the off-label population, which were divided into 4 populations. Less than 32 weeks of gestational age, again 40 weeks being full term, and postnatal age, less than 8 days. Group 2, less than 32 weeks gestational age and postnatal age 8 to 90 days. Greater than equal 32 weeks gestational age, postnatal age less than 8 days, and greater than or equal to 32 weeks, postnatal age 8 to 90 days. Okay, so problem number 1 was the formulation. The label dose for adults, or for people older than 90 days of age, was 20 milligrams per kilo. So if you have a premature infant weighing 500 grams, 20 milligrams per kilo times 0.5 kilos is 10 milligrams. We have a 500 milligram vial that now requires a 1 to 50 dilution, but the solution must be very highly concentrated to avoid fluid overload in these children. So this was problem number 1, is getting the dilution accurate so that the children were getting an accurate dose. Second problem is how to do a pharmacokinetic study. So a neonatal blood volume is 80 milligrams per kilo times 0.5 kilos is 40 mL. So this is a very, very tiny blood volume of this child. We minimize the blood draws by using sparse PK sampling and also using scavenged samples. So in other words, using samples that had been left over, we had times on them from when the samples were drawn, but they would be discarded, and so we use them to run the PK samples as well. The assays. So the goal of the drug dosing was to provide matching exposure and time over the minimum inhibitory concentration, or MIC, for each age group. And the goal specifically was to have the concentrations greater than 4 micrograms per mil for 50% of the dosaging interval, and greater than 2 micrograms per mil for greater than or equal to 75% of the dosage interval. And this is to cover for the gram-negative bacteria that would be covered by meropenem. The drug assays needed to be highly sensitive and accurate, using extremely small volumes of blood, less than 50 microliters. The dried blood spot method was developed and cross-validated with the plasma assays. So in the future, if people are doing pharmacokinetic studies on meropenem, they can just move to dried blood spots rather than having to draw blood. And it must be performed according to FDA's good clinical practice guidance because this data was going to FDA and the FDA would be auditing the labs. In terms of serious adverse events, these are right from the Code of Federal Regulations. Serious events include death, life-threatening adverse events, hospitalization or prolongation of existing hospitalization, persistent or significant disability, a congenital anomaly, or birth defect. But we were very interested in one safety question about seizures. The incidence of seizures in the study was 5%, but the question that we needed to know was, what is a baseline incidence of seizures of premature or any infant in the neonatal intensive care unit? And so we spoke with the people at Pediatrics who have contracts with many NICUs in the United States to find out what their baseline rate of seizures were in that population. And their seizure incidence was 5%. So this was extremely useful to have this database data as a background. Okay, in terms of the infrastructure that it took, the number of patients to perform this study was 200, so 200 premature neonates, and the number of sites, 24 sites. So in other words, to do this study, which was a relatively simple study, took 24 sites. So this is not, it's very difficult to do a single site study. So I just wanted to point out the timeline of basically 10 years here. So the written request went to the NDA holder from FDA in 2004. It was referred to NIH in 2005. We went through some negotiations with FDA in 2005. The request for proposals, the contract posting went out in 2005. The contract was awarded the following year. First patient enrolled in 2007. Last patient enrolled actually pretty quickly. It took about a year and a half to enroll all those children. The clinical study report submitted to FDA in 2009. The federal notice of the open docket, I will not go into any discussion about this, was in 2012. Labor negotiations between FDA and AstraZeneca took place at the end of 2014, and the label change was made in 12/24/2014. And this is the Meropenem label, which you can find on Daily Med, the National Library of Medicine's cache of drug labels, FDA drug labels. And you can see that there are differences in dose and dosage interval depending on the maturity of the child. Now, I'd like to talk now in terms of just some generalities about needs for pediatric drug development plans. Number one, you need a clearly defined question. You need a feasible pediatric study plan, availability of required resources, including site infrastructure. You need a study design team, a clinical team, a lab with very good facilities that can perform GCP lab quality assays, and you need a pharmacy. You need eligible pediatric patients. You need, if you can find it, the ability to leverage background and normative data so that you know what the baseline rate of the disease is in order to see that there's a change with your therapy, and you need good communication with the FDA. Okay, in terms of the feasible study plan, generally it's unethical to start a study that you know you won't be able to complete. There was a funny quote by a man named Louis Lasagna, who was a clinical pharmacologist, called the Lasagna rule. The incidence of patient availability sharply decreases when a clinical trial begins and returns to its original level as soon as the trial is completed. And you will find that this is the case. The other thing I'd like to mention is that there's an impression that the International Classification of Disease billing codes somehow equal the disease incidence or the number of eligible patients. This is not correct. So if you're trying to get an idea of how common the disease is, the ICD-9 codes will not be the way to go there. This is another indicator that studies are not being completed in pediatrics. So this was a really interesting paper called Clinical Research Involving Children, Registration, Completeness, and Publications. They identified all closed pediatric trials on clinicaltrials.gov, which had been funded by the NIH, and the resulting publications from NIH-funded studies from 2000 to 2010. And what they found was there were about 3,500 closed studies, but only 29% of the completed studies were published. And this indicates that there is a problem. There's a feasibility problem with these pediatric trials, which hopefully will be improved as time goes on. So again, in order to start your pediatric drug development plan, you need a clearly defined question. And just to use the meropenem case, does meropenem have an increased incidence of seizures compared with imipenem? And the sub-question is, what is the baseline incidence of seizures in neonates in the NICU? And how can we find that out? And the second question is, how are seizures diagnosed in the NICU? And does the FDA review division agree with the diagnostic criteria? So again, going back to the FDA and having a conversation about your drug development plan. The outcome measures, have they been validated? Are they agreed upon by the medical community and the FDA? If you were using a biomarker, it must be validated in your pediatric population. It doesn't mean that it's an FDA-approved, validated biomarker, but you should know whether the biomarker has been validated in children. Now, these are generally clinical trials for small populations. So it's important to use a clinical trial design for small populations, to consider published data as background or to incorporate pharmacokinetic priors for a pharmacokinetic sampling strategy. And it's also important again to have observational or natural history study data, if you can, as a baseline. It's important, if you can, again, to leverage database data. Not every question must be answered with a new clinical trial. Some data cannot be collected in one clinical trial, and database or other observational data can provide useful supplemental or normal value data. In terms of infrastructure, there are specific elements of infrastructure that should be included in your clinical study team as well as institution. You will need a pediatrician or a pediatric subspecialist who understands the complexities of the question you're asking. You will need a pediatric clinical pharmacologist in order to design the pharmacology aspects of your drug trial. You need an assay development team, a pharmacometrician to do the pharmacokinetic analysis, a statistician, a study nurse coordinator, and someone from your pharmacy department in order to make sure that the drug is available. In terms of institutional support, you must have institutional support to support your recruitment effort, to provide space for patient evaluation and procedures, data monitoring, regulatory support for FDA submissions, as well as regulatory support if FDA audits you. And as a piece of closeout advice, I found this over the years that when studies close, typically the materials for the study are all over the place. It would be hugely helpful to place all consent forms, case report forms related to inclusion-exclusion criteria, primary outcomes, and assay validation in one locked location for future FDA audit. In summary, pediatric drug development is complex. Each age group has its own complexities. The adult outcome measures may not translate into the pediatric population, and pediatric drug development requires a team approach and institutional support to succeed. And all of these are necessary to move from the former feeling that children's participation in research is not ethical to legislative and practical attempts to assure that it's not ethical not to. Thank you so much for your attention. This is Dr. Ann Zajicek. If you have any further questions, please feel free to contact me. Thank you.